Okay, well, I think we can go ahead and get started because I think we have those folks who are presenting first ready to go. Do you have a announcement slide you wanted to tee up, Kazan, before I share? Thank you. So today we're going to have a variant curation Q&A, which is kind of parallel to the gene curation one that we had a couple weeks ago. And so the variant curation proxies, and myself included, will all take turns presenting questions that people had added to the survey that we had sent around. We also hopefully will have some time to open it up for just questions from the group. And the next meeting will be on May 14th at 4 p.m. Eastern Time. The myeloid malignancy VSEP will present at that meeting. And then we also have just one reminder to fill out the ClinGen survey for AI and ML use and curation by today. We really just want to get a sense of what people are already using of artificial intelligence and machine learning as we really start to explore this within ClinGen. So we really would appreciate you taking the time to fill that out and inform our work. Great. Thanks so much, Kazan. I will take over. Deb, I think I'll have you share when you do your low penetrance, but I can share like the Q&A deck if we think that'll be simpler.
Sure, either way is fine.
Okay. Okay. So we're going to start the Q&A. Get my life together here. Okay, so for the agenda, we will start by answering the prepped ahead curation questions from that Q&A survey. And then Deb's going to take some time to talk about standardized text around low penetrance variants. And then we will end with an update on gnomAD v4 features and REVEL in the VCI from Christine Preston.
Great. Yeah, so I can get started. So we had a question on computational predictors. And so the question was the majority of computational variant effect predictors do not specify gain of function or loss of function. In many instances, this information is needed to be able to properly interpret computational predictions. For example, for a majority, if not all, oncogenes REVEL scores are above 0.65 for 50% of all possible missense variants. This is a very big issue, and ClinGen needs to stop pretending this problem does not exist. So here we are. I did put together some information on this and on REVEL scores. So if you want to go to the next slide. So just to orient everyone with REVEL and how a little bit of the sort of bones of how REVEL works. REVEL runs on a random forest model, and it's an ensemble-based meta predictor. So it uses the output of 13 other in silico predictors, and I've listed them here. So many of the predictors that it uses then, when it does this ensemble-based method, they as well rely on different types of information. So underneath in these programs, there's an empirical association with disease. So using germline or Mendelian conditions, usually from ClinVar or other data. Several of them rely on evolutionary analysis or sequence conservation, and some rely on physiochemical or amino acid properties and substitution properties. So all of that, though, is heavily informed by loss of function and by damage prediction. And so that's very different than change of function or evaluating enhanced function in variants. And so this causes some of the discrepancy that we see when you apply REVEL to known loss of function variants versus known gain of function variants as well. So if you can go to the next slide. So there are some caveats then for using REVEL with known gain of function variants, especially in hotspots. And so I wanted to point this out on the right-hand side where this is text from the publication, the initial publication from REVEL, where it talks about some of its the choices in the sets and in the predictive sets used. So one here says, finally, when given a genetic variant, and when a given genetic variant corresponded to multiple amino acid substitutions at the protein level, only one substitution was picked and at random. So you can see that if there are multiple substitutions that might be possible for a variant, picking one at random would be probably the least ideal for gain of function variants that might have multiple potential amino acids at that, particularly with something, for instance, like hotspots. which are associated with cancers. But it could really be problematic for any gene where one of the positions may have more than one amino acid substitution at that site, or where there could be, for instance, let's say two distinct diseases are affected by that variant, potentially distinct mechanisms. One of them might be loss of function, one of them might be gain of function. It could be benign for one disease or pathogenic for the other. So you can start to see that at any position where you would have multiple amino acids, the programs that are being used to inform that, it's a random choice that's being used at that position. So just to make everyone aware of that in the application of it. And next slide. So there is a recent article evaluating the use of REVEL in loss of function and gain of function variants. So I thought we could go through the results of this article and have that as some of our discussion around this issue. So in this article, the authors evaluated 131 pathogenic missense variants across these three genes. And the variants were classified into known loss of function and known gain of function variants. And it shows the number for each of these genes. And notably, there's another article where the same genes were evaluated in SIFT and polyphen, which are two of the programs that are used by REVEL as well. And it was also done by one of the same authors of this paper. And so these programs did also perform poorly on gain of function. So from this paper, they know these two programs performed poorly on gain of function. And then these two programs are also part of the input into REVEL as well. So if we can go to the next slide. So I put there's two figures from the paper, and I put two of them together. So this is just combining figures 1 and 2. So what we're looking at here is the REVEL score of the variants that they evaluated. And then as you're increasing in REVEL score, the number of those variants that are underneath this threshold. And the takeaway here really is that loss of function are more likely to meet the strong PP3 cutoffs, and these are the ones that are from the recent SVI paper, but they're more likely to meet this strong PP3 cutoff than gain of function. So you can see at the 0.75 mark, then actually I'm not able to see over the Zoom controls here. Let me decrease my size. At the 0.75 mark, about 89% of loss of function and 88% of gain of function are meeting this threshold, this 0.75 threshold. But as you increase, you can see a distinct difference in between the loss of function and the gain of function. Like in between 0.8 and 0.9, there's a really big gap in between those two lines. And so when you get to the strong level, the 0.932, then you can see that more of the loss of function are meeting that threshold than the gain of function. So that's just important to keep in mind. And I pulled the text here. This text on the right-hand side at the right-hand top is directly from the paper. So I think it's a good paper to look through if you are interested in evaluating loss of function versus gain of function for your gene. If we can go to the next slide. This is an example, and this is done by Dimitri Sonkin. So this is looking across the FGFR3 gene and looking at all possible amino acid positions and potential mutations in that, or potential variants in that gene, and then the REVEL scores. And so you can see that a high number of them all meet that threshold of at least 0.75 and greater. So this essentially just shows that it's challenging, especially I would say with FGFR3, to set a REVEL cutoff, especially due to the gain of function variants that are in FGFR3. So if we can go to the next slide. So, what I would say at this point is that if you know your gene has a missense gain of function variance, if you know that that is particularly involved in the disease that you're looking at, if you either have a known oncogene or disease mechanism that has a known gain of function, then it is appropriate to evaluate closely the performance of REVEL on those variants and to consider other predictors if needed. So, for example, FLT3 with the SC VSEP, so this is a somatic VSEP, ran multiple known oncogenic variants using the free tool Open Cravat and which provided multiple in silico prediction programs. And they found ClinPred had the best sensitivity specificity. And we are currently developing language to include in the variant SOP. So this is the draft text below that I'd like to get feedback on, where we would say that in silico predictive tools or missense variants can have known caveats and may require additional consideration. For example, missense variants known or suspected to cause gain of function, it may be advisable to assess the applicability of REVEL versus other appropriate predictors. So if you have additional language or thoughts to add on this, we are in the draft phase for adding this into the variant SOP. I think that was the last slide. Yep.
Great. Thanks, Deb. That was
Yep.
great. I will tackle the next one. That's about proband counting. This was specifically for PM3, but I'm just going to expand it to all proband counting on the next slide. So this person would like clarification regarding the use of PM3 and other criteria. So they provide the example if they're using PM3 for two siblings that have a variant of interest in trans to a pathogenic variant, can they count them both and also apply points for segregation? So let's just step back and clarify proband counting because this is a common question that comes up. So, to review proband counting in general, PS4 in the rare disease sense, because this is also used in case control settings for cancer or other common diseases with phenocopies. But in general, if you have a rare disease, PS4 is the autosomal dominant code, and PM3 is the autosomal recessive code for case counting. In the current Richards et al., it will change with the new version soon. So, but this concept still remains the same. So when counting cases, only probands should be used. Um, Not every pedigree will designate a proband. That's okay. You can designate a proband yourself. Oftentimes, it's really just randomly the first member of the family that comes to medical attention. So I think the more critical thing is only one individual per family should be counted towards PS4 or PM3. You really should not be counting multiple probands or multiple PS4 counts or multiple PM3 counts per family. If there are additional affected individuals in the family, they should count towards segregation scoring under PP1, because really that's just providing additional evidence of that family, not isolated individual observations of the variant in the population. So if we go back to the previous example, if you have two siblings that are affected and have the variant and a pathogenic variant in trans, which is a requirement usually for PM3, one sibling should be the proband and count towards PM3, and the other should apply towards segregation and apply to PP1 so that you're not double counting this information. Okay, I'll turn it over to you, Pam.
All right, so the question that I'm tackling is variants in ClinVar with minimal information. More specifically, if a variant has been submitted as pathogenic without much specific clinical information or reference for any experimental evidence, should we consider these variants for curation? And then furthermore, can these be categorized as a VOS or likely pathogenic? So this is I would say, common situation that occurs in ClinVar. And so I've put through a few scenarios to describe how to tackle this, especially from a ClinGen perspective. But one thing I just want to quickly say is it is important to do the due diligence. So if a submission has been put down as pathogenic, a variant submission, and there's no evidence to not assume that automatically it's a VOS or likely path without really um, digging further to determine if that is indeed the case. So if I go into the next slide, I thought what we would do quickly is just orient ourselves with an example of a ClinVar submission with no evidence. So here's a pathogenic, has been classified as pathogenic in mTOR, this frameshift variant, where you can see the condition that it's been curated, the context, who the submitter is, but there isn't anything else describing why they view this variant as disease causing. So in the next slide, I've got like the first scenario when you're dealing with the situation in the previous slide. There's a single submission, it's been put down as pathogenic, and there's no evidence supporting that classification. And one thing I first wanted to say is that in ClinVar, the reason why this is so common is that a lot of variant records are just based on a single submitter. And the figure on the right is data from ClinVar Minor earlier this year, just showing that the vast majority of submissions in ClinVar are just based on a single submission alone. And so what you can do in that context, if you want to find out more, try to understand the rationale behind why that submitter has thought this variant is pathogenic, is to reach out to them. And so ClinVar does have that information where they have the list of submitters, and you can just see that on the bottom left there, with general information and personnel and their emails. And from my experience with ClinGen VSEPs, there are a lot of submitters that are typically receptive and will provide more information. So that's something worth considering for cases where it's just a single submission. I've got scenario 2 in the next slide. So another scenario is when you have multiple submissions that are causing, and then there is conflict occurring. So in this example, there was 20 plus submissions for this one variant. So I've just shown a part of it. So most of those submissions were VUS or likely benign. And then there was one likely pathogenic submission that didn't have any data really explaining why it's likely path versus the VUS likely benign. And so we do have a process in place with ClinGen and ClinVar to deal with these type of conflicts, which in the next slide I will describe further. So this is done under the ClinGen Curation of ClinVar project, or CVC for short. So this is a joint workflow between the ClinVar team and ClinGen curators, where we essentially review submissions to ClinVar that appear inaccurate or outdated, and flag those submissions. And so the figure on the bottom right is just showing an example of what a flag submission looks like, where it'll have a little warning tag and the reason why it has been flagged. It's important to note that these submissions aren't hidden or they don't disappear, because they can still have valuable evidence embedded within them, or you may want to reach out to the submitters. So when we're doing this, another thing to clarify is that we're not necessarily checking the accuracy of the curation per se, but rather focusing on really obvious issues. So is the submission old? Is it an outlier? Is it lacking supporting evidence? And if it meets at least 2 out of those 3 criteria, then that's something that we would flag. And we're very communicative with submitters with flag submissions. They can respond back to us, they can add more evidence, and that would remove the flag. So taking it back to the example of a pathogenic submission without any evidence, and it's causing conflict. So if it's an outlier, it doesn't have that extra evidence, that's something that would be flagged and removed from the still present in the variant record, but it will no longer be adding to the conflict. And then in the third scenario, where you have multiple submissions and no conflict, which is something that wouldn't be tackled under the CVC project. So for example, multiple pathogenic submissions, and no evidence in any of them. And I think you typically would see this, for example, with a nonsense or frameshift variant. It might be helpful to then reach out to those submitters to see if they could provide evidence or their rationale. If there is a ClinGen VSEP, Variant Curation Expert Panel, with those specifications, you could also try to contact them as well. And I've just shown here a screenshot from the criteria specification registry. This is just showing the hearing loss VSEP as an example. So you can see, you can search for your gene and see if there are specifications in place, and that's something that the VSAP, if they have the bandwidth, may consider curating, or if they don't have specifications, but it's in their scope, perhaps they would consider including it in their pilot. One thing I just want to say in context of VSAPs and their overall bandwidth is in the next slide. So, sending a request to a VSAP to look at such variants is something that certainly can be done. And then I just wanted to have this slide just kind of flagging their overall priorities of VSAPs, which is to focus on those variants that would bring the most value to the broader community, whilst acknowledging their workload. It takes on average a couple of years to create specifications for various genes. And then there's also recurations that are done if, for example, a variant was to meet likely pathogenic or a VUS, then they have to recurate that every 2 years. So it is a bit of a balancing act, but they do focus on variants at medically significant classifications. So like path likely path versus a VUS likely benign. If multiple submitters have called the variant a VUS, reaching out to those submitters and combining the data can potentially push the variant into a benign or pathogenic state. Variants that can be put into a definitive state as benign or pathogenic because they don't typically require re-review. Variants that can be classified more quickly once the BA1, BS1, and PBS rules have been developed by the VSAP. And then, as I said, reviewing requests from the community. So I hope that answered that question. There's a few different scenarios and things to think about when there are variants submitted to ClinVar and they don't have the evidence to support the classification.
Thanks, Pam. There is a related question in the chat right now, so maybe I'll just turn you towards that while we're on this topic from Nazreen. When we want to compare the variants found in our projects with those reported in ClinVar and tag them as reported or not reported, which categories should be taken into account? Should we include pathogenic, likely pathogenic, or also VUS?
I think I'll need more detail as to the process you're describing there when you say, if you don't mind unmuting yourself and say your project and what you consider reporting. Like, it'd be helpful to understand more context there. Otherwise, if not, I'll just say generally speaking that, recommendation is that you shouldn't lean on what is in ClinVar alone for classification of a variant. And not to be biased, obviously, but with the ClinGen VSAPs, we have that FDA status because we do a very thorough analysis and review of variants before they're put into ClinVar. So barring that, generally speaking, I wouldn't say because a submitter has called something pathogenic, then that's something that you can then consider reportable in the context of your group. So it can help your analysis, but it's something to think about.
Yeah, the example is in cohort reports and papers. I will echo exactly what Pam said. In the clinical lab, we will, in our evidence summary, say that this variant is reported in ClinVar and include the variation ID. We don't say what it's classified as, we just say that it's present in ClinVar. And I would say, even for the same thing for cohort reports and papers, you can say that it's in ClinVar and include the variation ID. Just to let people know that it is there, but I would do your own classifications for variants. That's really why we got rid of those codes from the Richards et al. Publication that say like other labs classify this as benign or pathogenic, because you'd really evaluate the evidence yourself, especially in a clinical setting if you're returning things back.
Mm.
Great. Thanks, Pam. So our next question is, how old or recent should the information for curations be? And I'll shout out to Justine for answering this one, but she's under the weather, so I'm going to present it for her. So there's no requirement or restriction for curated evidence based on publication date. There are some factors to consider. For variants with many publications, maybe like really slam dunk pathogenic variants or something, scanning publications through time may alert you to a shift in the field's understanding of that variant. So if you're looking at really old papers that will say this is absent from 100 alleles or something, and it's disease causing, that's shifting from a proposed disease causing variant to a benign polymorphism with the most recent update of gnomAD, for example. You should also think about the classification of the variant. A likely benign VUS, likely pathogenic variant should really include all available evidence across time, because this hasn't necessarily reached a state of certainty potentially. A benign or pathogenic curation is not required to be comprehensive. The most informative evidence could be used preferentially, but contradictory evidence should not be ignored for that, just to get to like this state of certainty. Something else to think about is how well phenotyped case reports are. Earlier publications may have more detailed descriptions of patients, and more recent publications may include newer tests, which really could help meet your VSEP specification for inclusions of cases, particularly if they're saying they want exome or genome analysis or a very large gene panel to be considered. Probably those newer cases would meet those guidelines compared to some earlier, very old ones that might be well phenotyped, but might only have tested for one or two genes. And then something to think about, is this a new curation or a recuration? And if it's a recuration, a particular focus should be placed on identifying evidence made available since the last curation to see really if that's going to bump it over the edge, send it to a state of certainty, and make it the most up-to-date curation it can be. So I think at this point, because I do see that you're on, Christine, I'll turn it over to you so that Deb can really lead some more open-ended discussion after this, and then we can open it up for questions. Would you like to share the slides, or do you want me to
I'm
move them forward for you?
happy to share, Marina. Thank you.
Okay.
Yeah,
Yeah.
I just have a handful of slides. This should just take a few minutes. Thanks. Yeah, so I'm just going to give a brief update on some data that we're bringing into the VCI just to let everyone know. So as some of you are probably aware, we do have the in silico predictor REVEL in the VCI. We've had this for a while, but we did an update of the sourcing as of September 2024. And the reason for this was that the previous source that we were using for the REVEL predictor scores did not have the transcripts correctly associated with the scores. And there are instances which VSEPs were running into where the scores were different for different transcripts. So we wanted to be able to bring and display that information into the VCI. Can you click, please, Marina? Thank you. So to approach this, and also I'm going to talk a little bit about gnomAD in a second. In both cases, what we leveraged was the ClinGen linked data hub, which is managed and built by our colleagues at Baylor. And this facilitates efficient access to collated information, such as links and selected data from different data sources, and then they're made available. The VCI is using this, but I'll mention that the LDH, which some of you may have heard some really great talks from the Baylor team, including Neetu Shah. The LDH is available for anyone, so any group can use it. But the VCI is now sourcing REVEL scores associated with the transcript directly from the LDH, and I just have a really nice image that the LDH team put together here on the right-hand side. And next slide, please. Thank you. And so you can just get a sense of what this looks like in the VCI. Again, this is something that's already live right now in the current VCI. So this is an example where we had a variant that had different scores based on different transcripts. The transcripts are the ensemble transcripts, but we have indicated if they're main select or main clinical plus. So you can see that that's displayed here. We preferentially show the main select, and then you can expand it if you want to see other transcript scores. So that's the VCI, or sorry, the REVEL scores in the VCI. And the next slide, please. And then I wanted to mention an upcoming change that we're going to make to the population tab, which is we're going to be integrating in gnomAD v4.1 in the VCI. And this is coming out very, very soon. I'm hoping in the next few days, I'll be able to send folks an email to let you know what's coming. So the current data sources that we display in the VCI population tab are listed on the left-hand side of the slide. We currently display gnomAD version 2, EXAC, PAGE, 1000 Genomes, and ESP. We did an analysis, and many of those sources are largely or entirely subsumed in gnomAD, specifically in gnomAD v4. And so because of that, we've removed those sources so there wouldn't be double counting. And so when we do this release, then the upcoming data sources that are going to be displayed in the VCI population tab are going to be gnomAD v4. We're still going to display gnomAD v2 because we realize that some groups are still using that for curation. And we will also display the page data. And the next slide, please. And then this is just a screenshot of what the gnomAD v4 data is going to look like in the VCI. We're going to show the group max filtering low frequencies with the values for the exome and genome counts. That will replace the MAF table that we currently have in the VCI. And we're also going to display the genetic ancestry group table in the VCI for v4. The gnomAD version 2 data will remain as it currently is, and again, we'll also show page. And next slide. And I just want to take a moment to thank so many folks that we've had involved in this, especially from the Stanford Baylor team, Brian Wolf and Tiara Ferris and Neetu Shah. Oh, I see that Jenny's question about the hemizygotes for X-linked disorders. It's absolutely on our list. It's not going to be in this initial release, but that is something we plan on looking at very soon, Jenny, for the hemizygote data. Yeah, and that's all I have. Any other questions? Okay, thank you all.
Great. Thanks, Christine. I am going to stop sharing now. And Deb, I will turn it over to you to lead that discussion on low penetrance.
Sure thing. All right. Did we want to give a chance for anyone? So I saw, Dimitri, you had put a comment in the chat on the variants, the gain of function variants. And then there was one other question from Mayan about using the case table. Mayan, do you want to say any more about that question?
Well, sure, why not?
Hi.
You have the case table where you have summaries of all the papers that you gathered, and it gives you like a little three dots about whatever paper you gathered the information from. In our panel, it's important for the clinicians to see the information gathered from the paper and all the phenotypes that are seen, because for some of the genes that we're working on, they have specific phenotypes they want to see that are written in the paper. So when I need to present it, I need to go up each and every publication I used to go into that page and extract the information. In that table, you have the summary, but it doesn't show the summary. It just shows the first three letters, the first three words of it, and you can't extract it. I want like a button that you press and you can download a PDF of that summary so we can actually present that table to the panel and not go on each and every submission of papers that I've put on. Is that possible to do that? Because when I reached out to the people who are responsible for all the interface, and they said
For the
no.
VCI. So Christine is on, and she is one of the main point people for the VCI. So this would be, I guess, in a user request as an item. So this would be essentially for trying to enhance the functionality of the VCI for the use in the VSEP meeting itself. So this is in order to say you want to be able to render that table in a like a visual format that works for the VSEP meeting. Is that right? Okay.
Yes.
Yeah. Okay. Yeah. So I was just trying to get to an example of one, like so that I can see it myself. I'm not screen sharing yet, but I always like to also put my eyes on the question. I think I know. So when you're looking at the table, you're looking at like here's paper 1 and some of the evidence, paper 2 and some of the evidence. And you can see a little bit of the text there, but you can't really see it all unless you click into it, right?
Or mouse
Exactly.
over,
If
I
you want me to
think. Yeah.
share, I have one open so I can
Actually,
show
that'd be
everybody.
great. That'd be great. I'm like trying to scramble and find one. So yes, if you can find.
Oh, I need an approve, so someone.
You should be able to share now.
Great, thank you. And Here we go. So this is it's just specifically one paper. But
Okay, in
when
this
you're
table.
going to case segregation, you have this table. Usually you have multiple papers,
Hmm.
but you can see only the three first words for
Hmm.
the PP1 and for the PS1 and for the PP4. And I want to like a quick link so I can click on that and it will go out. Otherwise, I need to to press on that and I can't even copy it. It's not
Ooh,
able to be copied. And if I'm moving, it's gone.
okay,
So
so
I
you
need to
want
go
to be able
inside
to
this
expand it?
specific, I need to go inside here to go and copy all the information from here. So
Okay,
basically, when I have multiple papers, I have to do it for each and every one of them. And I don't want to do that. I want just a quick link.
got it. Okay. Well, I think that's a really clear request for it. And so if you were looking at that one that you just had up and you had the ability to expand that text right there on the screen so that you could see all of that text, would that work too? Or you want to just be able to copy that text to put it somewhere else?
If I'll be able to copy it from this table, it will be
Hmm.
great. But if I need to
Okay,
go inside each and every paper, it's like
got it. Okay,
it takes me forever.
okay. Well, yeah, I mean, Christine, no pressure to say yes or no right now, but we know that it's not a functionality that's current right now, but it seems to be a pretty clear request and maybe a potential possibility. So,
Yeah, I mean, what we can do and what we always do when we get requests like this is we can put together basically as a ticket with the request. Um, and then that will be considered as we go forward. So I absolutely appreciate you bringing this up. I'll capture the request. I will say that we're kind of looking forward to the V4 framework, and then that V4 framework is going to require a whole new interface. And so we can capture that request. We can consider it when we work with the proxies during software prioritization. But we're kind of minimizing our development right now on the VCI, what we're calling the V3 VCI, which is the active one, because we're doing a lot of work on the upcoming VCI. So I just do want to mention that. Um, but I really
yeah,
appreciate you bringing it up, and we can absolutely keep track of this request and consider it further. So thank you.
yeah, and I also
Thank
think
you. I appreciate it.
it's good to keep in mind that the functionality of this for the use in the VSEP review or for the use
Mhm.
in the VSEP discussions too. So I mean, I think I haven't thought of that necessarily in some of our discussions. I've been more like, how do I curate this variant,
Right.
looking at
Yeah,
it
that's a
rather
good
than
point.
like, how do we go further and use it in the VSEP? So thanks for bringing it up.
Mhm.
And I think just in general, this kind of usability of VSAP presentations is something that it's really helpful to know what the pain points are, because even if we don't end up fixing it in the current VCI, would really like to bring that to bear as we're doing the design for the upcoming
Yeah.
VCI. So in any case, your feedback is definitely heard and appreciated.
All right.
Thank
So
you.
yeah. So we can switch to, oh, so Dimitri, with your comment about the in silico predictors and assessing all possible missense variants. So you and I have had some discussion on this before. I do think that's a more challenging ask to have a research or to have curators, instead of taking the known and seen variants and evaluating those across multiple in silico predictors, asking them to create all possible missense variants at that position and evaluate that. So that's just kind of like vastly expands the task. So I think we'll have to have some more discussion on what we're asking curators to do there. I think currently, right now, all we're trying to do with this is to make sure people are aware of it, because that they're not kind of applying these missense predictors to known gain of function variants where it may be problematic. Um, I would say the next step would be to evaluate other appropriate tools to see if there's another tool that has better sensitivity, specificity for the known gain of functions versus either benign or versus other ones in the population, like maybe versus other ones in gnomAD, for instance. Um,
Right.
yeah.
So I think I wanted to mention, and the reason I'm mentioning in the chat is because in many cases, the variants which are known, they are very well selected. And
Yeah.
if one looks only at that variants, which is a mix of pathogenic and benign, it would look okay. But once you expand to all, the picture changes in a number of cases. And I think what I think is like, for example, alpha missense, which already provides the scores for all possible missense variants. And REVEL score is very similar to alpha missense scores in most cases. And also you mentioned the tool which already exists to look across many variants at the same time. So it's a little bit easier now to
Okay.
take a look, especially for something alpha missense, which already pre-calculated. And that would just give people the whole picture, because I want people to be aware, if you just look at the subset of well-described, for example, variants of ClinVar for a particular gene, you may not notice that there is a problem. Because essentially what's happening there, the reason why it essentially creates incomplete picture, because in reality, you're looking for majority of well-known variants, gain-of-function variants for gene. There is no more, essentially. You have 90% of them already listed there. So that's partially creates the problem when you look at only known variants and not all possible missing variants.
Yeah, you're working already with a very selected set. I understand that. So yeah, I think that in this, we can provide some discussion of that. It's always hard to provide notes about a thing without providing then further guidance of how to do it. So I think I would probably reach out to you to get some advice or guidance on further using, let's say, alpha missense, or how someone can just quickly run through that and do that in order to assess that should they have known gain of function genes. But yeah, so thanks for bringing it up, and thanks for the comments on it as well. All right. So I think that we'll switch to talk a little bit about low penetrance variants. So this is another topic in which we are trying to provide a bit of guidance. So standardized text is mainly the point here, but also to put a note into the variant SOP about handling and use of low penetrance variants, and as well providing information to biocurators about where they can find more resources if they're dealing with low penetrance variants. So let me pull up slides here and I'll screen share. So some of this we've already had in discussion with hereditary cancer biocurators, and I did do some updating on this based on a recent hereditary cancer call where we had discussed this. So let me put into slideshow. There we go. All right. So just to point everyone to this initial resource, so this is recommendations for the risk allele evidence curation, classification, and reporting from the ClinGen Low Penetrance and Risk Allele Working Group. So this was a publication that came out in 2023. Let me see. Okay, here. And figure 1 here provides an overview of the risk allele framework, Mendelian framework that needs future modifications for these low penetrance variants, and then high penetrance variants. And so this is typically the variants that we are curating, and the classification groupings here as well. So I do want to point everyone to a white paper that came out on the ClinGen website so far in January 2025, and the title is Current Challenges in Clinical Classification of Low Penetrance Variants. So this is from the Low Penetrance and Risk Allele Working Group. This has many considerations to review through if you are working particularly with a low penetrance variant, and especially in the cases where you're working with perhaps a gene that typically has high penetrance or typically has variants that are of high penetrance, and then you have 1 or 2 alleles within there that have lower penetrance as well. So I put the link in this slides, which I'll share after the call as well. So this initially was just trying to take a look at how VSEPs are currently classifying and summarizing low penetrance variants. So this was done in the fall of 2024. Just searched the ClinGen repo for these terms. So I used risk allele, low penetrance, different versions of this because currently when VSEPs are classifying these variants, they are not reporting it or putting it in the summary in consistent ways. Note that at the time I did not use moderate penetrance, so that was not searched. And if there are any variants classified with that, I did not pull those up. At the time, then I identified 25 variants that used this language. And so we're looking at, this is affecting a small number of variants overall, but the variants that are affected and for those groups handling it, they're problematic variants. So this is affecting 0.3% of the ClinGen variants at the time. And again, that's about 2,000 variants less than what we have now, or 2,000 or 3,000 now. So rerunning this again will probably pick up more. It affected 7 genes across multiple diseases that each of these had one matching variant. And here's the genes that are listed. And then 3 genes have greater than 1 variant. So these are the genes that have multiple variants that had this low penetrance or reduced penetrance language in them. And then I went through these to review how the VSEPs are currently noting this in the summary text. So the goal here was so that we can start to standardize how we are mentioning this in the summary text. Meanwhile, the low penetrance risk allele working group is working on further guidance about how to actually classify these variants in considering low penetrance conditions. So for VHL, you can see it meets BS1, so it meets this higher than expected allele frequency, but it has a reduced incidence or types of cancers. And then there were many carriers that were greater than our 60-year-old cutoff, but did not have VHL cancers. So overall, this variant had a decreased impact in terms that it had a decreased phenotypic impact. So it did not meet Danish criteria, which is a cancer impact that is used in VHL. For TP53, in this case, the variant did meet the threshold for the population, but in this case, there were many carriers with the variant above age 60 without cancers. There was no functional impact predicted in experimental assays as well. So what I wanted to do in this was to go through and look at some of the different ways and types that currently classified low penetrance variants, what information is causing them to be in this low penetrance bucket. For BRCA2, there was a higher than expected frequency in the population, but there was very strong evidence of meeting likelihood ratio analysis for family history. So they were showing family history of breast cancers, but it met PVS1, so it met a truncating that it truncated in a functional region, and it met experimental evidence, for instance. So it had that they classified it as likely pathogenic with reduced penetrance. For GJB2, it was another high frequency variant or higher than expected, but there was segregation data, and there was some experimental data. For F8, it's another higher frequency than expected, but reported in multiple cases. And then for USH2A, again, higher frequency than expected with segregation and with case and cohort data. So the takeaways from these, and I should say this was just, a kind of a quick look at some of the reasoning provided in the summaries, in the variant summaries. The takeaway is that the VSEPs have different reasons for noting a variant as low penetrance, and those reasons can include higher than expected population frequency combined with other significant pathogenic criteria, so including cases or strong segregation information. Conversely, it could meet the rare population cutoffs from the VSEP, but then display lessened or partial phenotypes, for instance, a lower incidence or a more or less aggressive cancers compared to the typical phenotypic impact of that gene and disease. It is the case that VSEPs are inconsistent in classification. So some classify it as pathogenic, but note the exception. Some classify it as a VUS, but note that it's a low penetrance variant. In all cases, though, the typical specifications that are used by the VSEP do not work for that low penetrance variant. And expert insight is needed to classify it appropriately. So there is no current ability to mark it as, let's say, an exception or to use the language that ClinVar has, which would be pathogenic, low penetrance, that is also modeled after the low penetrance and risk allele working group language that we saw from figure 1. So additional considerations to this is that we have had discussions in the hereditary cancer working group when we've recently discussed this as well about around issues of noting low penetrance versus moderate penetrance, assessing the penetrance in relation to the typical penetrance of that gene disease, how to assess this when there is an unknown penetrance of that specific variant versus if you have precisely known penetrance for that variant. So all of this is to say, the point here was so that we can start to draft some standardized text to handle the variants, as we know that there are variants already in VSEPs being classified and in handling this language. It's a very small percentage. So at this point, what we're trying to do would be to start to standardize the text while we know that further guidance will be coming out for the actual classification of low penetrance variants. So what we have so far and have drafted, and this has input through the hereditary cancer working group as well. So it would say, based on these PMIDs or references, this variant has been classified as, note the classification, and then a comment on the penetrance, if the penetrance is low, moderate, or atypical. And atypical would be used in cases where the penetrance is not exactly known, but it is atypical to the relative to the typical pathogenic variant in that gene, and then providing the rationale. And the rationale would explain why the variant presents as atypical for this gene disease. And that would then go into if it has high presence in the population compared to the typical variant, but still displays significant cases or segregation data that indicates it's pathogenic, et cetera. So I think I will stop there and have any questions or discussion and input on it. Know that if I'm seeing anything in the chat for it. So let me check.
Yeah, I don't think I see anything in the chat right now.
Okay. I know we've had great input and feedback from hereditary cancer coordinators and investigators as well. So if there are other groups where low penetrance variants are particularly problematic, it would be very helpful to get feedback on this too.
Deb, this is Jenny.
Yeah.
This is really great and very timely for some of the discussions we've been having in metabolism, not necessarily around low penetrance, but around these so-called pseudodeficiency variants. And I 100% agree that it would be really nice to have a way to flag these sort of unusual variants, I guess, in ClinVar. And I wondered if you had had any discussions with ClinVar surrounding how to flag those.
I haven't had that, but that was exactly the request that we heard in the hereditary cancer group, was just a way to flag this as unusual. This is an atypical variant for this gene. And I guess coming up with maybe atypical variant because it's pseudo defend like atypical variant, here's the reason
Right.
is kind of the request that I have been hearing.
Yeah.
Yeah.
Okay. Well, maybe we can join forces on that. And
Yeah.
I really like the guidance here, the classification, and then the low, moderate, atypical penetrance. We're
So
sort
would atypical penetrance work for the pseudo deficiency genes, or
of, I don't
would
think so.
different language be needed?
Yeah, I
Yeah.
think we've been thinking of sort of benign hypomorphic, but there's been a lot of
Mhm.
discussion about that. Some people like it, some don't.
Yeah.
Mhm.
So we're still we're still thinking about it. But yeah, I do feel like there's a lot of overlap between these hypomorphic or low penetrance variants and then some of the pseudo deficiencies. So we'd love to talk further. I know we just have a couple of minutes left, so I'll let other
Yeah.
folks go ahead. But yeah, maybe we can chat later.
Yeah, and I know hypomorphic is a term that often gets used in cancer as well. It does indicate low penetrance variants usually. So, but it's just even making sure that that terminology is used the same or consistently when
Mhm.
reporting it. So, yeah, I think it'd be great to discuss further and to see if there's one atypical flag that we could come up with some qualifications to it.
Yeah, that sounds
Yeah.
great. Okay, we'll be in touch. Thank you, Deb.
I think that was all the questions based on this. Is there anything else in the chat, Marina, that we want to go back to?
I think we've talked through a lot of them. Mayan stuck another request in the chat that actually came up in the discussion in ECT yesterday about alternate sources like DOIs, thesis links, such for PS3 specification. So we'll definitely take that back and think about that as we're developing the new VCI. Um, and I would say you had your contact information at the very top. So if anyone wants to provide any feedback in different disease areas for the reduced penetrance variants or atypical penetrance variants, that would be great. And I think we have 1 more minute. So does anyone else have any other last burning questions before we let you go?
Here, I
Okay.
I did, sorry, I did put the link in the chat. So I
Oh,
think
perfect.
somebody
Thanks,
asked
Deb.
for the link to the slides, but I'll also send the slides to you that they could be shared. I'm fine with that. So,
Yeah, I
yeah.
think you linked them in the other slide deck too. So
Oh,
we
great. I
have that.
did.
Yeah.
Okay,
Mhm.
good.
Yeah. Great. Okay. Well, thanks everyone. We are at time. Thanks, proxies. I think you did a great job answering all these questions. Great discussion, and we'll see you all in a couple weeks. Bye.